So welcome everyone, and thanks for joining this session on addiction, neuroscience and the quest for new treatments. Before I introduce our presenter, I wish to acknowledge I am hosting and recording this meeting from the lands of the Wurundjeri Woiwurrung people today. I acknowledge the traditional custodians and elders of the various lands on which you are living, learning and working on today, and any Aboriginal and Torres Strait Islander people who are participating. In the spirit of acknowledgement, I invite you to name the country on which you are joining us from in the chat box. So it gives me great pleasure to introduce Professor Andrew Lawrence. He is a NHMRC Principal Research Fellow and Division Head at the Florey Institute of Neuroscience and Mental Health, where he is a leader of the mental health research theme and runs the Addiction Neuroscience Laboratory. Andrew has published over 300 original articles and reviews and has been cited in more than 11,500 times. Andrew was Treasurer of the Australian Neuroscience Society and is an Honorary Fellow of the British Pharmacological Society. He was Senior Editor of the British Journal of Pharmacology and is currently Editor-in-Chief of the Journal of Neurochemistry and a Reviews Editor with the British Journal of Pharmacology. Andrew was recently President of the Asian Pacific Society of Neurochemistry and is currently a Council Member of the International Society for Neurochemistry and Junior Vice President of the International Society of Biomedical Research on Alcoholism. In 2017, Andrew delivered the Laurie Austin Plenary Lecture at the Australasian Neuroscience Society annual meeting. So welcome, Andrew, and I will hand over to you.
Thank you very much, and thank you for that very warm welcome, and thank you for the invitation to present here. I hope you can see my slides okay and hear me okay? Yep, good. Okay,
yes, we can.
So that as was introduced, the title is Addiction, Neuroscience and the Quest for New Treatments. So it won't come to any great surprise to you guys in the audience that alcohol use disorder and illicit drug use and tobacco use are huge problems. You look at the cost, this is the cost in the US. Alcohol itself outcosts cancer, tobacco hugely, more than double outcosts cancer. In terms of the prevalence and the cost to society in terms of money. And the terrible issue is that less than 20% of individuals with alcohol use disorder get any treatment whatsoever, and less than 10% get any medication treatment. In Australia, closer to home, alcohol hospitalises 430 Australians every single day, and alcohol kills 15 Australians every single day. That's yesterday, today, tomorrow, the next day. And that's a population of only 26 million people. So it equates to about somewhere between 4 and 5% of Australians having an alcohol use disorder. And at a global level, it's the single largest cause of preventable death in people aged between 15 and 49 years old. What are the current pharmacotherapeutics that are registered specifically to treat alcohol use disorder? There's obviously disulfiram, antabuse, that's been around since the early 80s. That's an aldehyde dehydrogenase inhibitor. Not on the PBS though. Naltrexone or Revia, which is an opioid receptor antagonist, again around since the mid 80s. And acamprosate, which has been around for almost 20 years now. And intriguingly, the actual biological mechanism of action of acamprosate is still not known. And the bottom line is these drugs work for some people, but at a population level, they are inadequate. They're simply not good enough. If we look at what's happening in a human brain from a heavy drinker in a functional MRI scan, you can see on the left here that when alcohol images are shown to social drinkers, low drinkers, we see activation of the ventral striatum or the nucleus accumbens. However, when you look at heavy drinkers, that activation shifts up into the dorsal striatum. And in fact, not only do we see activation of the dorsal striatum, but this activation correlates with what's known as the obsessive compulsive drinking scale score. And so we can see that the desire to consume alcohol is directly related to the activation of the dorsal striatum as opposed to the ventral striatum in heavy drinkers. Oh, excuse me. So we asked a question, well, what's happening in terms of molecular adaptations in the striatum, specifically in the dorsal striatum, associated with long-term alcohol use? And so what I'm going to do is I'm going to take you through a paper we published just 12 months ago in Biological Psychiatry, where we've identified what we believe is a valid novel therapeutic target for alcohol use disorder. And so first thing we did is we accessed some alcohol and control postmortem brains from the New South Wales Tissue Resource Centre. Now, this part of the brain bank in Sydney is curated by NIAAA. It's considered the cleanest, highest quality alcohol brain bank in the world. These brains were from alcoholics that were not smokers, that didn't have cirrhosis and didn't have hepatic encephalopathy. You can see that subjects are matched for age, for BMI. There's a slight mismatch on postmortem interval, but importantly, the brain volume to weight ratios are not different. The brain pH is not different. And what we call the RNA integrity number, or the quality of the tissue, was not any different. But you can see a huge difference in terms of alcohol intake, the number of drinks per week, some people up to 500 drinks per week, and the number of years spent drinking. So we took those brains and we took the striatum, and here you can see in this cartoon the caudate being the medial part of the striatum and the putamen being the lateral part of the striatum. That will become important as the talk progresses. And we just did complete genome-wide RNA sequencing of the striatum for these human brain samples. And we were very conservative. We decided to look at the 300 most regulated genes with a significant large fold change. And as you can see from this circle here, here's the 300 top most regulated genes. About 170 were upregulated and about 130 were downregulated. If we did non-biased pathway analysis on that data set, G-protein coupled receptor signaling came out as the #1 impacted pathway, and the #2 pathway was cyclic AMP mediated signaling. Not surprising, because many GPCRs signal through cyclic AMP. And what we saw when we actually looked at individual G-protein coupled receptors, we saw a number of receptors that didn't surprise us at all. Sort of GRM5, this is the metabotropic glutamate receptor #5. DRD1, that's the dopamine D1 receptor. GPR88 is an orphan receptor that's been linked to alcohol use disorder, and CNR1 here, which is the cannabinoid CB1 receptor. We were expecting those, so that didn't surprise us at all. One thing that we did see that we weren't necessarily expecting was CHRM4, which stands for the muscarinic M4 acetylcholine receptor. So that really struck us as something quite interesting that we decided to pursue. So the first thing we did was we wanted to confirm that sequencing data. And we did that by a technique called RT-PCR. And to do this, we separated out the caudate and the putamen from these samples. And you can see from the graph here that there was actually no alteration of any of the expression of any of these receptors. So the cannabinoid receptor, the muscarinic receptor, dopamine D1, D2, D3, or GPR88. None of these were regulated at all in the caudate. However, when we went to the putamen, which is that lateral part of the striatum, implicated in habitual behavior, we see that we replicate almost entirely that data from the RNA sequencing. So then we went from the mRNA level, or the gene level, to the protein level. So we set up a binding assay in the same caudate and putamen tissue samples, and we found that indeed in the caudate of these brains, there was no change in muscarinic acetylcholine receptor binding whatsoever, either the total binding or that we ascribed to the M4 receptor. However, in the putamen, we saw a downregulation of muscarinic receptors, and that could be described or ascribed completely to a downregulation of the M4 muscarinic receptor. So we see a reduction in the expression of the gene, a reduction of the mRNA encoding that gene, and a reduction of the protein in the human brain. So the next thing we wanted to do was to see if we could actually recapitulate this in an animal model so that we could interrogate more closely what was happening. So we took some rats and we gave them 5 months voluntary chronic intermittent access to 20% ethanol. They also always had water available at the same time. So they were not coerced into drinking, it was completely voluntary. And they had access on an intermittent basis about 3 or 4 times a week. And we had 3 groups of animals. We had those that were age-matched ethanol naive, those that had had 5 months, which is around about a quarter of the lifespan if you're a rat, that had this intermittent access paradigm. And then we had another group that had had the same ethanol access, but then were withdrawn for 14 days afterwards before then we took the brains and we sampled the dorsomedial, representative of the caudate in the human, and the dorsolateral, striatum representative of the putamen in the human. And then we did hypothesis-driven qPCR to look at those specific receptor genes that we found in the human. And what we saw, so here's just showing you the drinking data. These animals, they gradually learn to consume or start to consume alcohol. And this is grams per kilogram per day. So they're averaging about 7 grams per kilogram per day. So it's a pretty heavy intake over this time, but it's always voluntary. You can see they've always got the ability to choose between water and ethanol. And then we punched out samples from the equivalent of the caudate and the putamen here. And what we can see is that we see very little action in the medial striatum, a little bit with the D1 receptor, but we see basically we recapitulate essentially almost all of what we found in the human brain in the dorsolateral striatum, the cannabinoid receptor, the muscarinic receptor, and GPR88. Remember, we didn't recapitulate the effect on the D1 receptor. We weren't expecting to see anything in the D2 receptor. That was there as a control. That didn't change in the human either. So we almost entirely recapitulated the human data set in a rodent model. And here you see, in particular, the muscarinic M4 receptor was being modulated, particularly more so, and the cannabinoid receptor and GPR88 for that matter, in the withdrawal group. And that is important because if you think back to the human tissue, those humans would have likely gone through periods of drinking, withdrawing, periods of drinking, withdrawing, periods of drinking and withdrawing. And as you'll see later on, we think those withdrawals are actually very important in terms of this dysregulation of the expression of this receptor. So we wanted to interrogate this with some pharmacology and some functional behavioral experiments. So we managed to obtain this molecule VU telephone number, which is what's called a positive allosteric modulator. And it works specifically and selectively at the muscarinic M4 receptor. The VU stands for Vanderbilt University. That's where our collaborators are in the US. And this is just some in vitro pharmacology to show to you that it is what's known as a positive allosteric modulator. That means it binds at a site on the M4 receptor that's distinct from the site where acetylcholine binds to. But what it does do is it potentiates the effect of acetylcholine. So as you can see here, it's enhancing the affinity of acetylcholine for the muscarinic receptor when it's present. And you can also see from this graph that it's also increasing the efficacy of acetylcholine at the M4 receptor. So this compound is only going to be working and only going to be doing what it does when the presence of acetylcholine as well. It's potentiating the effects of acetylcholine. So we wanted to find out if this drug was any good at reducing or interfering with self-administration of alcohol. So we took animals, we trained them to self-administer alcohol for around about 60 days. They had always had the choice of either water or alcohol. And then after they were stably self-administering alcohol, we dosed them up with this compound or vehicle. And as you can see, we gave it to them orally to try and imitate what would happen in a human around 2.5 hours prior to testing based on the pharmacokinetics of the drug. And we tested them for the ability of this drug or vehicle to impact on alcohol self-administration. And then 2 days later, the animals went in and were tested again with the reverse. So the ones that had vehicle first time got drug, and the ones that got drug got vehicle in a counterbalanced manner. And here you can see the group data here and a time course data here that these animals in a 20-minute session for operant responding, you can see they're pressing a lever around about 130, 140 times. They have to press 3 times for every alcohol delivery, and you can see, and they consume everything that's provided to them. And you see from each individual rat at a group level, this drug is quite effective at reducing the desire of these animals to obtain and consume alcohol. Has no effect on their desire to obtain and consume water. And if we look at the alcohol lever on a time course basis, you can see in the vehicle treatment, the bulk of the action is in the first half of the assay where they really load up on a big binge and then tail off. And we can see we really reduce that with this drug. We did a full dose response on this drug, and we found that the 10 milligrams per kilogram dose, half a log unit lower, had a trend, but it was not significant. And the higher dose at 100 milligrams per kilogram was actually marginally sedating, so we were getting non-specific effects. But we can see we do have a window of efficacy where we can reduce alcohol self-administration by targeting the muscarinic M4 receptor in this way. We also found, doing a slightly different test, on the previous test, every time they wanted to obtain alcohol, they just had to press that lever 3 times and alcohol was delivered. Here we did a progressive ratio. It's really designed to test the motivational strength of alcohol and whether this receptor is modulating the motivational strength. And so under this paradigm, each subsequent delivery of alcohol costs more effort. And so the lever press requirement goes up and up and up and up. And so they get to a point which is a break point. And the break point is the measure of the number of lever presses that they're prepared to do to get a single dose of alcohol. And you can see here under vehicle conditions, once we ask the rats to press 30 times for a single shot of alcohol, they decide that that's not worth that much. And you can see over a time course, again, as we showed you before, the VU compound significantly reduces that motivational strength or that desire to go out and seek alcohol and then consume it. And by the way, these are all specific to alcohol. We did analogous experiments allowing animals to consume sucrose, and it had absolutely no effect whatsoever. I'm just not showing those data because of time constraints, although they are in the publication. And in fact, sorry, here is the sucrose data, my mistake. You can see absolutely no effect whatsoever on the same dose of the same drug in the same cohort of animals that are trained to, a separate cohort of the same group of animals that are trained to self-administer sucrose as opposed to alcohol. So it is quite specific. We wanted to check that the 30 milligrams per kilogram dose, as well as it being specific to an individual reward, wasn't impacting any other functions of the animal, particularly motor functions that could impair, or could cause reduced responding. So to check that, we trained rats to walk on a rotor rod. This is a rod that's spinning around. They stand on it, and the first day we habituate them on a fixed speed. It's going around at 4 revolutions per minute for 2 minutes. We treat them with vehicle. 2.5 hours later, we give them 2 trials. The next day, we do the same thing, but the rotor rod is accelerating from 4 to 40 RPM over 2 minutes. But we do the same vehicle treatment and then 2 trials interspersed with a 15-minute wait in between. And then on the third day, it's an accelerating rotor rod. We treat them with either vehicle or the drug, and then do the test twice over with a 15-minute break in between. And here you can see the data, absolutely no effect whatsoever. It has no capacity to impair the animal's motor coordination or ability to walk on a rotor rod. So we know that it's not impacting their ability or their desire to consume sucrose or perform the task to obtain sucrose, and it's not causing any ataxia or motor sedation whatsoever. As you know, relapse is a big issue, so we figured next thing we should do is test whether or not we can impact upon relapse-like behavior with this compound. So here's the paradigm. We trained animals to self-administer alcohol, again, 60 days, as we showed you before, and then we put them into rehab, what we call extinction, where the alcohol and the cues that were signaling alcohol availability are no longer present. So when the animal presses the lever, there's no consequence. And what happens is over a period of days, their responding on the active lever disappears because they undergo new inhibitory learning that under this new contingency, pressing the lever has no consequence. And so the behavior becomes very similar to that on the inactive lever or the lever that was previously delivering water. When they get to this point in time, we treat them with either the drug or vehicle and give them a reinstatement test. And what we do on the reinstatement test, this is just to measure seeking behavior without the impact of the consumption on top of that. So the way we do that is we reintroduce the cues that previously signaled alcohol availability back over here when they were learning and consuming alcohol. But when they press the lever, the cue is illuminated, for example, but there's no delivery of alcohol. So they're pressing in an association with the cue, which is a measure of the drive to seek out the alcohol. After that test, we take them all the way back to reacquire self-administration, back through, extinguish them a second time, and then test them again in this manner in a counterbalanced way. So each animal is its own internal control. Half of them are treated with the drug on the first test, half of them are treated with a vehicle on the first test, and on the second test, they get the alternate treatment. And here we see the data. So here's the extinction responding on the active lever. You see it's very similar to what the animals were pressing on the inactive lever. it's gone from about 140 or so, as you can see, down to about 10. When we inject them with vehicle and reintroduce those cues that were paired with alcohol availability, we get this reinstatement of alcohol seeking, which is the akin of a relapse-like response. When we treat them with this compound VU that's targeting the M4 receptor, we can see that at the group level, and you can see all the individual data points, that we have a substantial effect at reducing that behavior. Doesn't impact the latency to respond at all, again, so another indication that it's not causing any motor impairment. So if we go back to this fMRI image that I showed you before, remember, we're talking about the activation of the dorsal striatum, and if you think about the molecular data that I showed you, it seemed to be that most of the action was happening in the dorsolateral striatum or the putamen in humans. And so what we did is, to test that out, we went back to another group of rats and we targeted, because in the past, all of those experiments I showed you were targeting giving drug systemically, which means the drug has access to muscarinic receptors wherever they are in the brain. What we're doing here is we're injecting the drug directly into the dorsolateral striatum, so that this is the only place that the drug can be acting, just to test whether that's a site of action. So we did surgery, animals were trained to self-administer as before, and then we did surgery to implant cannula into the brain, specifically into the dorsolateral striatum, so we could inject the drug directly into there. And we tested them under the self-administration contingency and the cue relapse reinstatement contingency. And here you see in the self-administration, the group data and the time course data, we get a significant reduction in alcohol intake. It's not a dramatic effect, but it's, as you can see over a time course, it's a significant effect, particularly when they're doing that early binge-like behavior. Importantly, however, it almost completely prevents the relapse-like behavior. And remember, this relapse is happening after they've been through extinction, where they're not getting any alcohol. So at this point in time, they've been withdrawn from alcohol for somewhere between 10 and 12 days. And this test also is in the absence of alcohol, whereas this test is in the presence of alcohol. So you can see we get a very dramatic effect of this ability of this M4 positive allosteric modulator to dramatically reduce relapse-like behavior. So and that's just a mapping of our injection sites, showing the dark blue and the light blue spots being the self-administration behavior, and the dark blue spots being the relapse-like behavior. So to summarize this part of the talk, hopefully I've demonstrated to you today that muscarinic acetylcholine receptors, particularly the M4 receptor, are regulated by chronic alcohol use in the dorsolateral striatum of rats. And I haven't shown you today, but it's in the paper, that is actually happening on the dopamine D1 neurons in the striatum. I've shown you that this compound, this VU compound, which activates M4 receptors, selectively and specifically reduces alcohol self-administration and cue-induced relapse-like behavior in rats. And so we believe, based on that combined human and rodent data, that these represent novel therapeutic targets for the treatment of alcohol use disorder. In fact, 4 or 5 pharma have got active programs on this particular target. They believe it's going to be a novel antipsychotic, but we think we've got another indication for them. Okay, second part of the talk, I'm going to shift gears a little bit just to show you some translation that we've been doing. So over 15 years ago now, we were the first people in the world to link the orexin system, which is a neuropeptide found in the hypothalamus, to alcohol seeking. And this is in situ hybridization histochemistry showing expression of the gene encoding the orexin peptide in slices of rat brain. And this is a non-preferring rat. This is a preferring rat. And here you see on the right-hand side a preferring rat that's been allowed to consume alcohol for a considerable period of time. And I think what you can see is that the individual cells that express this peptide, the density of expression in an individual cell is not changing. However, what we're doing is we're recruiting expression in cells adjacent that normally express very low undetectable levels of this peptide. After long-term alcohol use, they start to express significant levels of this peptide. We interrogated this system pharmacologically, showing you again this cue-induced reinstatement. This is the alcohol lever, this is the water lever. You see we inject the animals systemically with vehicle. They reinstate, they're responding on the alcohol lever. And we inject them with a selective orexin receptor antagonist, and we can completely prevent that behavior. And again, there's no effect whatsoever on the water seeking. This was the first evidence in the world for a role for this peptide system in cue-induced relapse behavior. And further, we went to show that, again, we can discriminate between alcohol and a natural reward. This is going back to that progressive ratio analysis, looking at the motivational strength. And with a very low dose of this drug, we can reduce the motivational strength of ethanol without having any effect whatsoever on the desire to obtain and consume sucrose. So we think that, again, that this is actually a very good target for alcohol use disorder. And just to show you that, we've actually got a phase 2 trial currently underway at St. Vincent's Hospital. This is the NCT number. This is the title, suvorexant in the management of comorbid sleep disorder and alcohol dependence. It was suspended due to COVID-19, but we're back recruiting again, fortunately. I'll just finish off again with another potential therapeutic target. We think phosphodiesterase inhibitors may be useful for drug and alcohol abuse, mainly because they're implicated in cyclic AMP signaling that I showed you was strongly implicated in our human postmortem data, plus they're anti-inflammatory. Here you see some data that we published close to 10 years ago now with rolipram, which is a PDE4 inhibitor, reducing alcohol self-administration, reducing the preference for alcohol, again, not reducing sucrose self-administration. Another group did something similar with a phosphodiesterase 10A inhibitor, but you'll notice here that they reduced both alcohol and saccharin self-administration, so it wasn't specific to alcohol, whereas we found the PDE4 inhibitor was. And here we see ibudilast, which inhibits both PDE4 and PDE10. Again, selectively reduces alcohol intake. I'm only showing you the alcohol data here for the interests of time. And there was a phase 1 trial on ibudilast for safety, which has been completed. The results were not posted. However, the same group are now recruiting for a phase 2 double-blind trial with this NCT number here. Ibudilast and the PDE4/10 has also passed a safety trial for methamphetamine use disorder, and also has been shown to selectively reduce the subjective effects of methamphetamine. And this same group that published this a few years ago now, 5 years ago now, under this NCT are recruiting a pilot study for the effect of ibudilast on inflammation in methamphetamine users. So I'd just like to finish off by acknowledging my colleagues who worked on the muscarinic story that I told you at the Florey and at Monash and at Vanderbilt. I'd like to thank the New South Wales Tissue Resource Centre and the NIAAA for the human brain samples, and the NHMRC for funding the research. And I'd just like to leave you with a quote from Edgar Allan Poe, which is a perfect demonstration of the fact that not everyone uses drugs of abuse for pleasurable reasons. I'll read it out to you. I have absolutely no pleasure in the stimulants in which I sometimes so madly indulge. It has not been in the pursuit of pleasure that I have perilled life and reputation and reason. It has been the desperate attempt to escape from torturing memories, from a sense of insupportable loneliness, and a dread of some strange impending doom. A very telling quote, I think you might agree. I'll leave it at that, and I'll be happy to take questions if we have time. If not, I'd be happy for you to collect some email questions and I can answer them offline at any time.
Does anyone have any questions? We've got about a minute. So I'll just open it up to the floor if anyone does have any questions. No? Okay, if you do, feel free to email us at VADA and we can pass them on. I have just put the link. Firstly, I would like to say thank you, Andrew, for your presentation. That was most informative. And also, I would like to say that I have the link to the next session, our final session, which I've just posted in the chat. If you do want to join, that session is the pathway towards drug checking in Victoria with Dr. Monica Barrett. This session I'm going to keep open. There is a 10-minute intermission between now and the closing the last keynote. That's just so you've got time to grab the link and also take a quick break. Yeah,
if anyone wants to stay online for the next 5 minutes or so and ask me a question, I'm more than happy to do that.
Fabulous. And you can also do that as well. So we'll see you at 11:40 over in the next session. And as I said, this one will stay open for about 10 minutes.
Thanks, Caitlin.
No worries, Andrew.